Hi class, for this discussion we are going to talk about the nervous system. And our nervous system is beefy. So we're going to take out this discussion and break it up into 3 separate lecture recordings. Hopefully no individual lecture recording will be longer than 20 minutes. This first recording is going to focus primarily on an overview of the nervous system. Our second recording will focus on the central nervous system, the CNS, and our third recording will focus on the peripheral nervous system and the drug disorders associated with the nervous system as a whole. So let's go ahead and dive in. Let's talk about how we can break up the nervous system. We have the 2 main divisions of our nervous system, the central nervous system, CNS, and the peripheral nervous system, PNS. And it's very important to enunciate your words when you say PNS, otherwise your students snicker at you. So if we look at our central nervous system, it's going to be made of the brain and the spinal cord. Anything outside of the brain and spinal cord, by definition, is going to be in the peripheral nervous system. So any nerve, any ganglia, which is kind of a pre-processing center outside of the central nervous system, and any sensory receptors is going to be part of the peripheral nervous system. As we look at these 2 main divisions, CNS, the central nervous system, was the brain and spinal cord, and then the peripheral nervous system gets to be a little bit more complicated. We have the sensory division, which sends information to the brain and spinal cord, and we have a motor division, which carries information away from the brain and spinal cord. Within our sensory division, we have the somatic sensory division. This is going to be what we consider classic sensory information, and then we have the visceral sensory division. This is going to focus on sensory information from the organs of the abdominal pelvic cavity. So this somatic sensory division is typically going to be associated with decision making, voluntary decision making, that kind of information. The visceral sensory nerves are going to send the brain information that's involved with maintaining homeostasis in our body. When we look at the motor branch of our peripheral nervous system, this is going to carry information away from the brain and spinal cord. And there's 2 primary divisions of the motor branch, the voluntary and involuntary. The somatic motor division is going to send information to skeletal muscles. The autonomic, or automatic, as I like to think of it, motor division is going to send information to smooth muscles and cardiac muscle tissues. So it's going to be involuntary in nature. And within that autonomic branch, we have 2 different kinds of involuntary responses. One involuntary response is going to help us to run away or to fight our way to freedom. So this is going to be associated with high stress situations. This is the sympathetic division. And we also have a division of our autonomic nervous system that helps us relax and recover. This is known as the parasympathetic division, and it's commonly called the rest and digest division of the autonomic nervous system. Within this nervous system, our nervous system, we're going to have lots of rapid communication. That's a hallmark of our nervous system. Quick, rapid communication, the orders of fractions of a second up to a second. So we'll have rapid communication between the cells. We'll collect information, process the information, and then make a decision and act on it. So we'll have sensory input from the sensory branch of the PNS. We integrate the data in the CNS, the central nervous system, and then send the signal to the motor branch of the peripheral nervous system to act on our decision. The 2 primary types of cells within our nervous system are neurons and the neuroglia. Neurons are the rock star. They're the one that send and receive the electrical signals, and the neuroglia are the support crew. The neuroglia help the neurons be the best possible neuron they could be. When we look at our neurons, we'll spend most of our time focusing on the neurons because they're the rock stars or the divas of the nervous system. These neurons are going to be categorized based off function, location, and shape. So there's different ways to categorize them. We can categorize them based off of the function. So we have sensory neurons. Sensory neurons are going to carry information to the CNS. Motor neurons carry information away from the CNS. And then interneurons process information within the CNS. So information in, information processing, and then acting on the information. Think back to chapter 4 when we talked about different cell types and different tissue types. If we look at a typical neuron, and I'll just talk about these terms as I look at this figure. A typical neuron is going to have a cell body, also known as a neurosoma, and then there'll be many branches that come off of it. Most of these branches are going to send information to the cell body, and those branches that send information to the cell body are called the dendrites. And then there's going to be one long branch that carries information away from the cell body of the neuron or the neurosoma. That one long branch is the axon. As we look at the axon of many neurons is going to be myelinated. This myelination, this myelin that covers the axon of many of our neurons is a lipid-based covering. It's 80% lipid, 20% protein, and it covers the axons to make it so that we can send electrical information faster. It speeds up nerve impulse conduction. How does this do this? Myelin insulates, it's electrically neutral, so it can insulate the axon. And one other important feature of our myelin sheath is that it guides the regeneration of the axon within the peripheral nervous system. So when we have nerve damage or damage to our nervous system in the peripheral nervous system, we can regain function as the axons guide, or as the myelin sheath guides the axons back to where they need to connect. This myelin sheath is made of a very specific kind of neuroglia known as the Schwann cell. This is a neuroglia that's only in the peripheral nervous system and myelinates the axon. In between myelination sites, I'm just going to flip over here. So here we have a typical neuron. This typical neuron has a myelinated axon, and you can see individual, so 1, 2, 3, 4, 5, 6, 7 individual Schwann cells wrapping around the axon to myelinate it. And there's little gaps in between the Schwann cells where there is no myelin. That gap is a Neuron, and I'm pretty sure I'm mispronouncing that. It's a French word. I apologize if you're French and you know I'm mispronouncing it. Those Neuron are where the actual electrical signal is conducted. So that electrical signal, as it's moving down the axon, jumps from node of Rainway to node of Rainway. And this jumping motion of the electrical signal is known as saltatory conduction. Salta is the Spanish word for jump. As we, excuse me, sorry about that, I hit the mic there. As we are maintaining a neuron at rest, we're going to need to have the ions, the electrical ions in the neuron maintained so that it's primed and ready to send an electrical signal across. This nerve impulse or electrical signal is known as an action potential. And we start with the resting electrical voltage. This resting electrical voltage or resting potential of a neuron is the voltage across the membrane when it's not actively conducting a nerve impulse or action potential. And at resting potential, we have more positive ions outside than inside. So the inside has an overall negative charge because there's more positive stuff outside of this axon than inside. So if we look here, in particular, the two ions we're going to focus on primarily are sodium ions and potassium ions. And at rest, we have lots of sodium outside and lots of potassium inside. So here is the resting membrane potential. We have more sodium out of the cell, more sodium or more potassium in the cell, and more overall positive on the outside. The inside has an overall negative charge to it. Now we can start conducting our action potential. This action potential is going to be a rapid wave of depolarization, or you could think of it as an electrical current traveling across the membrane. Sometimes you can think of it, some students like to think of it as ions moving back and forth across the membrane of the axon. When we start the action potential, we need to open the sodium gates. By opening those sodium gates, sodium ions rush in with their concentration gradient and make the inside of the cell more positive. This is called depolarization. So the inside of the axon is going to lose negative charges because we're adding positive charges to it. So we'll go from negative 55, which is what opens the sodium gates. These sodium gates open at negative 55 millivolts, and it will go all the way up to positive 35 millivolts. So we become much more positive inside the axon. At positive 35 millivolts, by increasing that voltage, we can then cause sodium voltage gated channels to open up. This causes repolarization. As positive sodium ions leave the inside has less positive charges and becomes more negative and goes back to that negative 70. We'll have waves of depolarization and repolarization traveling down the axon. First a wave of depolarization, which is immediately followed by a wave of repolarization. And at the very end, afterwards, all the ions are put back to where they started with a sodium-potassium pump. This is a special pump that uses up ATP to move sodium and potassium ions against their concentration gradients, thereby restoring the resting membrane potential. So here we can see The axon that's reached threshold is a negative 55 millivolts, and at negative 55 millivolts, the sodium voltage gated channels open up, and sodium ions start to rush out of the cell. Excuse me, rush into the cell. I misspoke there. So the sodium voltage gate channels open up at negative 55 millivolts and sodium rushes into the cell. And then at positive 35 millivolts, our open sodium channels close. Oh, I'm getting ahead of myself here. And these potassium channels should open up and potassium rushes out of the cell. Here is a phenomenal YouTube video that you should watch that illustrates this process. I'm not going to click on it because if I have a YouTube video within a YouTube video, my video usually gets taken down for copyright purposes. But you should definitely download this PowerPoint and click on this video. As we go from a neuron to neuron, we need to have a special connection between those neurons. And that connection is called a synapse. A synapse is the small gap in between the neuron and the receiving cell. And in most cases, that receiving cell is going to be another neuron. The sending neuron is referred to as the presynaptic neuron. It's the one before the synapse. The receiving neuron is the one after the synapse. It's the postsynaptic neuron or postsynaptic membrane. As we are going to transmit information across the synapse, we need to go from an electrical current to a chemical mode of information. And this chemical that we use is going to be a neurotransmitter. There are many kinds of neurotransmitters. Acetylcholine is the most common one, but we also have dopamine, serotonin, histamine, ATP, nitric oxide. I could go on and on. There's over 30 identified neurotransmitters in our body. Acetylcholine, dopamine, and serotonin are some of the more popular ones, or more commonly talked about neurotransmitters. And these neurotransmitters are stored in the axon bulbs of the axon terminals and can be released into the synapse when the axon is electrically stimulated. So how does this neurotransmitter get into the synapse? We'll have that electrical signal, that nerve impulse, reach the end of the axon terminal. At the axon terminal, calcium ions start to rush in when it becomes electrified. Those calcium ions rush in and bind to the storage vesicles or the synaptic vesicles and cause them to fuse with the cell the presynaptic membrane and exositize the neurotransmitter into the gap. So the wave of depolarization goes down the axon terminal, calcium ions rush in, bind to the axon, the synaptic vesicles. Those synaptic vesicles fuse with the presynaptic membrane and exositize their neurotransmitter across the gap. The neurotransmitter diffuses across the gap and then binds to a neurotransmitter receiver, which opens up and lets more ions move in and out of the cell. We can have synaptic integration occur. And the synaptic integration is when we have either an excitatory signal or inhibitory signals be received by the same neuron. Some neurotransmitters cause the cell membrane to become more positive. Those are excitatory signals. Some neurotransmitters cause the cell membrane to become more negative. Those are inhibitory signals. Because neurons receive excitatory and inhibitory signals, they need to process these signals at the cell body. And this is where integration occurs. If we have more excitatory signals than inhibitory signals, we will send the information down the adjacent axon. If this neuron receives more inhibitory than excitatory signals, it will not send a signal down its axon. More excitatory signals can move the neuron up towards the threshold voltage to initiate the action potential. Inhibitory signals can move it down and away, and excitatory and inhibitory signals are added together. So we can have a positive charge and a negative charge canceling each other out and being represented by this black line. So, class, concept checked. If a patient was given a medication that caused their sodium channels not to open up, it blocked those sodium channels, how would that affect the nerve impulse or action potential? Would it speed up the action potential? Would it slow down the action potential? Would it have no effect on the action potential, or would it block the action potential? This is a thinker question, so go ahead and ruminate on it for a bit and then get me an answer. 5, 4, 3, 2, 1. The correct answer is it would block the action potential. These sodium voltage gated channel blocking medications are oftentimes called painkillers, particularly anesthesia. is most often going to function as a sodium channel blocker. And when the patient is given anesthesia, their nerves lose the ability to send information down the axons within the nerves, and they lose sensation. The difference between anesthesia, which puts you to sleep temporarily, and euthanasia, which puts you to sleep and then kills you, is the dosage. Euthanasia, particularly if you think of taking your pet, your cat or your dog to the veterinarian to have them put down, is a very high dose of anesthetics. And if you have a high dose, you cause so many of those sodium voltage gated channels to be blocked that your nervous system shuts down completely and you die. And that's all we have for this lecture recording, part 1 of the nervous system. If you have any questions for me, please feel free to post them on the class discussion board, shoot me an email, or stop by my office. Happy studies!